Here today. It's a pleasure to be able to introduce this topic of what is cancer and what is metastasis. I'm Dan Welch. I'm a professor in the Department of Cancer Biology here at the KU Cancer Center. And I'm going to give a brief history of what we know about cancer. And I have turned off, sorry, we'll start again, but I have turned off my speaker and it's coming through here. So I don't know where it's coming from, but it's not playing here. So, okay, sorry about that, everybody. We're working out technical bugs. You'd think after 4 years of doing Zoom, we'd have it all figured out, but not the case. Anyway, I'd like to thank everybody for being here today. I'm Dan Welch, a professor of cancer biology and one of the associate directors here at the KU Cancer Center. I've been asked to talk about the topic of what is cancer and what is metastasis. And what I'm going to do is a very quick overview of some of the history of what we've known about cancer and a little bit about where we're going, and finally ending up with where you as patient advocates might be able to play bigger roles in our process of discovery and treatment of cancer patients. So this slide may surprise you, but cancer is something that has been known about for over 4,000 years. The first written clinical trial, in essence, was a papyrus published in ancient Egypt called the Ebers Papyrus, and it actually talked about surgical removal of what we know now as cancer, chemotherapy of cancer patients, where they ground up the periwinkle flower, and giving patients, I'll affectionately call it scum from the Nile, to induce a fever, so immunotherapy. So back 4,000 years ago, we had 3 general classes of cancer treatments. And I think that's a pretty impressive thing to realize that before they wrote it, they'd actually studied it enough to write this stuff down. Many people don't know that the first recorded mastectomy, a modified radical mastectomy, occurred in 30 AD. The daughter of King Darius of Syria was treated with a mastectomy. I highlight that because they didn't have anesthesia back that far. So these patients underwent a great deal in order to try and be cured. We've obviously added quite a bit in the intervening years, and the most recent new class of therapies, cellular therapies, or what you may have heard as CAR T cells, were really tested and treating beginning in 2017. And as this time has gone on, I just gave a brief list of several of the things that have happened. I just want to highlight that our thinking about cancer has evolved. Back in Egypt, they thought about where this mass occurred, in which tissue it occurred. But with the development of the microscope, people could begin to look at the cells in those different tissues and noted that different cells and different tissues gave to different types of cancer. And as the cells began to be understood more, we looked at markers on their surfaces and things. So in this case, estrogen receptor and progesterone receptor in breast cancer, or androgen receptors in prostate cancer, and so on. These began to be markers that are being used currently in our treatments of those types of cancer. About a decade or 2 ago, people began looking a little bit more at the molecular genetics of the cancer and looking at mutations that occur and different genes that are turned on and off in the cancers, and then combining all of that information for these molecular profiles. So our thinking is evolving even to 2024, which is when I'm recording this. So I think that's important to know. So we know that cancer exists, but let's go back just a little bit further and ask the question, what is cancer? And about 20 years ago, 2 of the leaders in the cancer research arena, Bob Weinberg and Doug Hanahan, proposed what they called the hallmarks of cancer. And we'll walk through those very briefly. They are self-sufficient. They're like an autonomous car. And I think you know from the Tesla experience that if you have things that are too autonomous, they make mistakes. And cancer cells do that. They resist growth inhibitors. This is the picture of the world's tallest man. He was over 9 feet tall. And the world's shortest adult man. He was under 2 feet tall. Cancer cells resist death, much like Wonder Woman here. And they're immortal, like Yoda. Cancer cells sustained the recruitment of new vessels to supply them with nutrients. That's a process called angiogenesis. And like Harry Potter here with his cloak of invisibility, they evade the immune system so that they're not being attacked by the immune cells. They have metabolic alterations. I like to think of myself as somewhere in between these two cats on the top panel, but they're not at the middle of the road in terms of their metabolism. And cancer cells are genetically unstable. So normal cells in our body generate a mutation about once out of every billion cell divisions. That's billion with a B. But cancer cells have been measured to generate mutants in as little as 1 out of every 10 cell divisions. So that genetic instability leads to new cancer cells that have different properties. And we'll cover that in just a couple of minutes here. And then the hallmarks of cancer is that they promote inflammatory responses. They get the immune system to come in to actually help the cancer cells do what they're going to do, including invading surrounding tissue and metastasizing to other tissues. So if I put all of that into one slide, cancer cells have all of these different hallmark characteristics. If this is just a list of what I just showed you, but I want to point out one new one that was published just last week in the journal PLOS Biology. And it shows that cancer cells can actually eat or cannibalize the tissues in which they're growing, break down that tissue, and use that as nutrients for the cancer cells themselves.
So are you guys going to do that next week then, you and Marcia?
Okay, whoever's online needs to mute themselves, please. Thank you. And my friend, Bob Libby, took this picture in his garden last week, and it's of a dandelion, and I hope you can see these two slugs that are inside that dandelion. They're eating the dandelion for their own nutrients, and he happened to have thought this was a great analogy, and I did too. So while I've taken a lot of the pictures you'll see in this talk, I give credit to others who've done so. Okay. As these hallmarks of cancer have been characterized, it's been increasingly recognized over the course of the last 15, 20 years that it's not just the cancer cell we have to worry about, it's the host interactions, the immune cells that come in, the attempt of the body to wall off the cancer by building a wall around it. Other characteristics are plasticity, which basically means adaptability. The cancer cells change in response to that environment, including the bacteria that invade the cancers, and the bacteria in our gut controls the behavior of cancer cells in other parts of our body. And so the use of probiotics and prebiotics are beginning to be used more commonly as part of the whole cancer treatment regime. So you may ask yourself, what are the causes of cancer? And there are 4 basic categories: genetics, our lifestyle, the environment we live in, and sometimes just bad luck, that genetic instability. So that leads me to a terrified look on the people who are in this room. I'm giving you a quick pop quiz. What percentage of patients' cancers are due to inherited mutated genes? Less than 1%, 5 to 10, 50, or 99? 5 to 10. We have a winner. I'll have to find something to give you as a prize. But yes, but if you talk to the average person on the street, they'll say, well, my cousin had it, therefore I'm more likely to have it. That is a common fallacy. And it's really important to know that we can only define 5, 10, maybe 15% for some cancers. So that is something I think we need to pay very close attention to, but it's not the only thing. The environment contributes just as well. So what causes the cancer? Well, it takes the first step is these normal cells, which behave in a predictable way. They do functions that they're programmed to do. But then the first steps are that they become immortalized and genetically unstable. So those cells begin to divide. As they divide, they generate new cells that carry these mutants, and they go through typically a benign phase in which they just grow. They're not controlled, but they're not going elsewhere in the body. Malignancy is defined as the ability of those cancer cells to begin invading and breaking through what's known as the basement membrane, which is like a wall, and they break through it. And that defines malignant cancer. And then some of those invasive cancer cells can acquire the ability to spread to different sites. So just being locally invasive is not the same as being metastatic. Okay. So with that, you saw that we've evolved our treatments. And I want to briefly just address the key question. Why is cancer so hard to cure? Well, so far, we've really not identified characteristics of cancer cells that are not present in our normal cells at some level. They just do what they're doing in the wrong time, the wrong place, and by design a therapy to target a behavior of cancer cells, that's going to lead to what are called dose-limiting toxicities, because if we're treating the cancer cell, but it's inhibiting the ability of cells to crawl, well, our immune cells have to crawl. So we end up having a lesser immune response because of that effect. Or we have off-target effects. Drugs are not perfect that target only one thing. If they have something that's close to it, you can affect those things as well. So this lack of unique differences in cancer cells creates a difficulty in treating them. Another aspect is what's called tumor heterogeneity, and I'll define that in just a second, but it's also this genetic instability. Because they're genetically unstable, every time cancer cell divide, the children of those cancer cells are not exactly the same as their parents. And what that leads to is this heterogeneity. And here I've depicted just this cancer. And blue would be a benign part of the cancer. The red is the malignant part, okay? And cancers are defined by a pathologist by the most dangerous looking cell in a biopsy. So if you have one invasive cell, the whole cancer is called malignant. But if you don't see any invasive cells, you call it benign. So how could this heterogeneity make it more difficult to treat? Let's just say the slide that we're analyzing in the biopsy is this blue line A. Well, it only cuts across this region here, and we would see only benign. And this would be misdiagnosed as a benign tumor. Down here, we would diagnose it correctly because it would have red cells in it. D, depending upon if there's a red cell in it, we may or may not. So it's important to be able to recognize that that's a problem in our understanding of what the cancer is doing. But by far and away, the most lethal attribute of cancer cells is metastasis. And that's, in fact, what I'm going to spend most of my time talking about here. But here are just some statistics from recent studies. In breast cancer, if it's diagnosed at stage 1, 5-year survival was very close to 100%. But if it's diagnosed with metastases already, it's under 20%, 5 years. Now, those numbers are changing and getting better. Patients are surviving longer with a good quality of life, but they are never considered cured at this point, and we need to change that. I'm going to be a bit political for a few seconds here. The first ever count of the number of women living with breast cancer that had metastasized occurred only 5 or 6 years ago. It is not measured in any of the cancer registries anywhere in the world. And now that women and men are living beyond 5 years, this is, I think, a critical issue that you as advocates can help take up to get our agencies to count these people, because they deserve the treatment and follow-up so we understand what's going on. Okay, that's a political statement, but that's why we're here. Another thing is, just so that you can help be advocates as well, 90% of cancer-related deaths are due to the process of metastasis or spread to different sites. Still, less than 5% of funding in the EU, the National Cancer Institute, the Chinese Cancer Institute, or the Japanese Cancer Institutes goes towards research on projects that focus on metastasis. So there's a big disconnect in what's killing people and what we are funded to study. So again, you can help with that. Now, it's not to say any of that research that's not metastasis isn't important, but I do think there's a good reason to advocate for a higher percentage. All right, so now how do these cancer cells metastasize or spread? And I've drawn a number of animations, and I've asked, as you listen to this, don't look at any of this and try and memorize it. Just sort of get a picture of what's happening, because you can always ask. I'll be more than glad. You can email me anytime to answer questions. But I think if you just get a flavor for what's going on and how complicated it is, I think that'll be very important. So the first step is the tumor has to grow. And it's genetically unstable. It generates heterogeneity. And some of those cells will accumulate enough mutations that they will be able to metastasize. Some will not, but at least some will. And before the cancer cells move anywhere, they start communicating with the rest of the body. They send out signals in these little sacs that contain molecules in them called exosomes. And those sacs go to the bone marrow, and they cause mobility of the immune system. And the immune system cells go to different organs, and they modify those organs so that they create sort of a vacation home for a cancer cell that's eventually going to go there. And I'll show you a little bit more of that in just a second. But so this cancer cells, they grow, they mutate, they begin communicating with the rest of the body, and then they acquire the ability to migrate or crawl. 20 years ago, people thought crawling. How many ways are there to crawl? Well, humans crawl in different ways too. Okay, cancer cells have different ways of crawling. Okay, and they're all effective for different reasons. And in the era of cancer, or the area of cancer, we have 4 different types of motility. And I'll just quickly talk to you about them. Collective motility is when cancer cells move as a cluster. I jokingly say, but I think you can all relate, this is like teenagers at the mall. They move as groups, and they disrupt things as they move through. Okay, they can move as individuals, as this amoeboid activity. In this case, the cancer cells just squeeze through little places, or they can actually convert between cell types. They go through what's known as an epithelial to mesenchymal transition. Don't worry about the big words, but they can do that. And then the last one, I just want to show you this. It's pretty rare, but it's very freaky. You can have cancer cells passing through the middle of another cancer cell and coming out on the other side. That's known as entosis. It was first described for white blood cells exiting the vascular system or a vessel, and they pass through the cells lining a vessel and exit on the outside of the vessel. Okay. Amazingly, they don't kill the cell that they go through. Okay. About 5 years ago, a colleague of ours, Adam Marcus, showed something that there are leaders and followers in cancer cells. And he's kindly shared this video with me. Come on, video. It's not going to show, apparently. Ah, there it is. So if you look up here, this leader cell, these others follow. Then the leader moves off, and these other followers are lost. They don't know what to do. So they're actively looking at what is this leader cell, and maybe if we can control that, we can stop this process of motility. Okay, that's again relatively new information. I have not heard if any characteristics that are distinctive yet, but it's obvious what's happening, and he saw this in multiple places. I just want to show this. If you look at these orangey figures in the top, this is collagen. And why do you care? Well, collagen is the structural component of a lot of your tissues. And this is all exactly the same molecule, but in different tissues. And collagen is organized differently in each tissue. And cancer cells have enzymes that can break down collagen, but they break down this collagen only if it's organized a certain way. So the enzyme that breaks down collagen in this tissue, doesn't necessarily break it down in this other tissue. And so they've specialized in such a way to be able to do that. And as they're breaking down that collagen, they're taking it from being a sort of mesh here, like a chain link fence, and they're organizing it into parallel sheets. And what this does, as you can see here, it's a little more crisscross, parallel. You start seeing the cells moving at a slow rate, but after they line that collagen, they move about 100 times faster. So the pioneer cells that leave the tumor in the beginning create an environment so other cells can leave more rapidly. They do this also by interacting with the nerves that are coming into a cancer. And those nerves are sensing pain and so on. And it turns out the nerves are controlling a lot of the responses, including the recruitment of inflammatory cells. And this is a macrophage that's migrating into this cancer. And then the cancer actually reprograms the macrophage so that those macrophages change and they align with the tumor cell. So it becomes a tumor cell alternating with macrophage. So tumor cell, macrophage, tumor cell, macrophage, tumor cell, macrophage. Can you hear it? They form a conga line. And that conga line is where the macrophages are actually doing the invasion for the cancer cell. And to put it into an analogy for those of you like the Kansas City Chiefs, go Chiefs, the front line breaks a hole so that Pachenko can run through. Okay, he's got to do a lot of work, but first they create that hole. These macrophages are doing that for the cancer cell. And they do this in coordination with the cells lining vessels, the tumor cell, and the macrophages. And this is called a tumor microenvironment of metastasis. And there are teams of investigators looking at how to break up this little coordinate and prevent the cells from ever escaping the primary tumor. And as they get to this blood vessel, which is how they'll spread most of the time, as the cancers are dividing behind them, they're doing like this Japanese subway in which they actually pay people to shove more folks onto the subway to get them to fit there. Well, if you think of the subway as the vessel, the cancer cells that are dividing behind it are pushing other cancer cells into the bloodstream. And that's a process known as intravasation or entering vasation. And the cells get in and they roll along. They're still maintaining contact. And a couple of little facts that you can bring up at the next cocktail party that you go to. Between 1 million and 4 million cells a day from a pencil eraser sized tumor enter the bloodstream. If you believe the statistics that we have cancers that are around for 10, 20, 30 years before we can detect them, that could mean billions of cells have started this process of metastasis. The good news is it's highly inefficient. The bad news is it happens. But the cells that get into the bloodstream are known as circulating tumor cells, and several groups around the world for the past 30 years have been characterizing those circulating tumor cells. Here are just a couple of pictures, just so that you can have an idea. That process I mentioned earlier in the epithelial mesenchymal transition. Here you can find that cells are totally epithelial, totally mesenchymal, and everything in between. So it's been a challenge to find them in the bloodstream and characterize them, because out of the billions of cells that get into the bloodstream, less than 1 out of every 100,000 at best complete metastasis. So which ones are the right ones to study? We don't know. Okay? The cancer cells cause the blood to clot around them. It kind of puts a cage around them so that they're not being easily killed. And that helps them to evade the immune system. They also are changed by the platelets and the white blood cells that are sending signals, so that the cancer cells find, besides evading the immune system, they get turned on by the immune cells. Thank you, Michael Jackson, for this video. I still think it's a cool one. But unfortunately, a couple of people who are younger than I don't realize that was a Michael Jackson black and white album. But I think, illustrates how we're all very similar, but we can just shift a little bit. In the bloodstream, a couple of fun little things happen. Blood is flowing, and in the veins, it's flowing a little slower, but you have shear. But in the arteries, there's a lot faster pushing. And so the shear is a little bit stronger, okay? And the good news is a lot of the cancer cells get ripped apart during that time. But we know that a lot of them get ripped apart in the heart itself, but during this transit, well over 90% are dead within an hour after they get into the bloodstream. Okay. Getting into the bloodstream and surviving is one part of it. They also have to get out of the bloodstream. They can squeeze through, much like this octopus. It can go through a space much smaller than itself, and it gets recruited by that immune cell that we talked about earlier that had migrated from the bone marrow to the different tissues. That has created that vacation home, or what we call the pre-metastatic niche. The cancer cells land near where those immune cells have gone before them, and they are more effective at growing and colonizing that tissue. There are many studies ongoing now to try and understand exactly what that interaction is between that pre-metastatic niche and the tumor cells. I think it's important to know that getting out of the blood vessel is very inefficient. But then some cells begin to proliferate. They take over the tissue around them. And some of those cancer cells actually form tubes themselves, and blood will flow through cancer cell-lined vessels. Still haven't figured out how that happens, but it does. All right, sorry, another pop quiz. Breast cancer that spreads to the bone is now bone cancer. If you listen to CNN, Fox, CBS, NBC, and so on, 9 out of 10 times they will say, well, she had breast cancer before, but now she's got bone cancer. Or he had prostate cancer, now he's got bone cancer. No. Important, you can help educate people that wherever the cancer started is what kind of cancer it is. It's not behaving the same in those other tissues, but it is still the tissue of origin. All right, so several of you are familiar with something called a Venn diagram, and we have these hallmarks of cancer, but not all cancer cells are metastatic. So my colleague and I, Doug Hurst and I, define the hallmarks of metastasis as these 4 characteristics. Don't necessarily have to remember them, but if you think of the black here is the Venn diagram of cancer, white is the Venn diagram of metastatic cells, and then where the metastases occur can form the magic 8-ball Venn diagram of metastasis and where it occurs. And we need to understand all 3 levels of this. Still a lot to do, but there are groups looking at genes that promote metastasis. There are a ton of those. I'm not going to really deal with it. My lab's been focusing on the class known as metastasis suppressors. And metastasis suppressors are basically the brakes of a cell, and they get turned off. The brake line doesn't work anymore. And I'll just show you one example and how that's relevant. Here we have a melanoma that does not express a metastasis suppressor we call KIS1. And if we inject those cells into a mouse, you can see there these green, we've engineered them to be green. They grow, and within 5 weeks, this poor mouse is dying. If we have KIS expressed in those exact same cells, they get to the, this is lung, same rate, but they sit there as single cells for months. If we take those lungs where the single cells are, we bring the cells back out, they still grow, they still have the KIS, they look fine, they still express KIS. We inject them back in, the melanoma, it grows just like the parent cell, and it goes back to the lungs. So what does this tell us? It says that they've completed every step of metastasis except growing at the secondary site. And if we could control that, that's a good thing, because all those steps prior to growth have occurred before a patient is diagnosed. And what we are working towards is understanding how KIS might be used as an anti-metastatic treatment. Okay. So our good friend Cheryl Jernigan likes this analogy. I've updated a little bit with Mr. Ed, but what we want is a horse that's in the barn, trapped in the barn. What we have is rampaging horses. Well, what KIS might offer is an intermediate. The horse is not in the barn, but it's under control in the pasture. And what this may allow is patients to live with a cancer being controlled as opposed to being completely cured. Cure is best, but good quality of life and longer life would be very good. Recent studies are looking at what we call metastasis modifiers. Again, this analogy I may have to change because a lot of people no longer know what this left pedal is. It's the clutch. But the clutch are the modifiers. It changes the ratio and the transmission. And these are things that change efficiency of metastasis. And we and others are looking at genes that are the baseline or the genetic background, or perhaps the race, if we look at it in that way, and how that could explain some of the differences between different races and so on genetically that explain why some patients develop aggressive cancer and some do not. There are plenty of other socioeconomic reasons for disparities, but there are genetic components. In the last couple of minutes, I want to talk about clinical trials for anti-metastatic therapies. This comes about from a grant that we had with PCORI, the Patient-Centered Outcomes Research Institute. And I want you to come away with a bit of understanding of what it takes to think about therapies. Again, this is not meant to be memorized in any way, but just to give you a flavor. Okay. What should we consider when we are designing a trial? What is the target we're going after? What will we consider a successful outcome? How would we measure success? Okay, is it measured by the patient, the caregiver, both, the regulatory agency? And what are our barriers to that success? And is our treatment feasible? I will give you a little anecdote. When I used to work in the pharmaceutical industry, a clinical colleague wanted to do a sabbatical in my lab. And for 6 months, he did spend time in the lab. And I told him one day, this is what I think we could do. And he says, that is absolutely brilliant, Dan. But no nurse in the world would ever do that. And no patient would ever accept it. Unfortunately, he was right. But it was an important lesson for me a long time ago. Okay, so what is the ideal target? Well, we've already talked about there's nothing unique that we know of, but it's got to be accessible. It doesn't do you any good if you can't get into the vault at a bank to get the money out of the vault. So we need to deal with that. So some of the trial-related challenges that occur, the time to get a result. If we can't measure something happening in 4 or 5 years maximum, then nobody can afford to do the trial. It's billions of dollars already. It would be trillions. There's a set of criteria to measure for whether a treatment works, and it's called RECIST. It won't deal with it too much, but that idea of keeping cancer stable would be considered a failed therapy because the cancer hasn't shrunk. And we need to see if we can revise those criteria to look at different outcomes. The complications, there's not a pharmaceutical company in the world that tests against metastasis in the preclinical trials. They tested against primary tumors. They're not the same. And there's a big regulatory burden to be able to do the animal work and the clinical work. Patient-related issues, you all very well aware of these, but patients are typically not involved as much in most institutions. We do better here than most places in writing consent forms in a language that everybody can understand. We require patients to go to get the trial instead of having the trial come to the patient. That's a big hurdle, especially for people who are lower income or whatever. Enrollment is typically, in clinical trials, is typically only after they've exhausted everything else. And that should not be the case if we can avoid it. And then all of these therapies cause major toxicity. You as patients or your caregivers want you to have the highest quality of life possible. You want a lot of long life, but if it's a miserable time, that's not ideal. Everybody deserves access and as minimal impact on their livelihood, work life, just doing life and enjoying life. Whatever side effects occur, we need to take care of those instead of treating them after the facts, try and be aware of them and prevent them in the first place with palliative care. Detect earlier, and I was very pleased when Jill Peltzer and I led this PCORI conference. Patients wanted more information about why metastases occur in different organs as a priority. Bone mets don't kill people, but they make lives absolutely miserable. Understanding bone mets is a critical thing. Brain mets kill people. So they have different, they're both bad, but they have different outcomes and we need to understand it. So with that, I have a handful of take-home messages. Cancer is many different diseases. It's really complex, but we're understanding it and our understanding is evolving and we're every day we learn something new, but it's not enough today. We'll know more tomorrow, but that's still not enough. And I hopefully have just given you a bit of extra encouragement that you as research and patient advocates are critical to this process. You can influence the direction and the pace of research. And most importantly, you increase the relevancy of whatever research we're doing. So with that, I give a big thanks to a number of advocates with whom I've had the privilege of interacting in my lab. And several of these are from Pivot, others are from around the country. But I learn every time I talk to each of you. Hopefully, I contribute a little bit to you learning a little bit from me too. And with that, I thank you, and I will be more than glad to answer questions if I can.
Peggy had a question. How many women are diagnosed in BC of the 1,154,000?
I'm sorry, I was echoing here.
Danny, it's Peggy. I'm,
Okay.
you showed a picture of 150,000 women and men that were metastatic in the United States today. Do you have any inclination on how many of them were diagnosed metastatic to begin with? De novo.
Actually, that information, I don't know that number.
Okay.
But that information is actually in the SEER databases and in the state registries. If one is diagnosed with metastatic disease, that is recorded. It's the ones who develop metastases after that that are not recorded because it's not typically in the oncologist's office, it's the primary care doctors and so on. That is the primary issue.
Okay. I'm curious about that. I'll just see if I can't find it somewhere. Thanks, Danny. It was a great presentation. We appreciate your time.
And I see some chats, so I can read them. So Tanya was reading yours, but it was echoing in my computer, so I apologize. Oh, excellent. You were answering the questions online. Thank you. Yeah, I appreciate the positive encouragement. If you have other questions, feel free. I don't see any more in the chat, but well, I was either totally clear or I confused everybody. Oh, wait, new message. Thank you, Sylvia. It was a little longer than I'd expected, but hopefully that little extra bit about the trials information was provocative and thoughtful for you.
Danny, this group probably really appreciated the information on the clinical trials because we're all patient advocates and trying to impact that. So everybody, I'm sure, appreciated the extra time.
I just feel compelled to say this. In the past month, I have heard a former postdoc of mine was diagnosed with metastatic kidney cancer. And two other friends were diagnosed with one with breast cancer and another one with testicular cancer. And I got an email yesterday from a pastor who was asking for help in navigating the health system. So it hasn't directly, I haven't had cancer, right? I don't, but I want you to know that it's everybody here at the KU Cancer Center. I can speak for everybody, and they won't mind this. We've all been impacted by this, just as you have. But it hurts. It still hurts, and we want to save our friends as much as you want to save yourselves and your friends. So, anyway.
I have a question. Can you hear me, Danny?
Yes.
Okay, I'm in my car, so I just had a question. Is from my understanding with my cancer, is it the same with blood cancer? So I had primary CNS non-Hodgkin's lymphoma, diffuse large B cell, but that was also found in lymph nodes in my body. So I'm assuming that that is someone like what you were talking about with the breast cancer, that would be a stage 4 where it's found in other areas, that
Yes.
same cancer. Okay.
Yes. Lymphoma is a blood cancer, but it's not. It's a bone marrow tumor that
Right.
actually colonizes a lot of different places. Lymphoma is one of those weird things. By the way, I have a couple of folks from my lab who were sitting in here along with a couple of other Pivot colleagues. And I appreciate Tanya understanding that I really wanted to have people that I could see. It's not the same as on a computer screen where I can't see anybody. But one of the things I tell all of our graduate students when I'm teaching, or the medical students, everything I said today is right 90% of the time. That other 10%, we just have to memorize, and we have no clue of why it's that way. Cancer doesn't play by all the rules. It's a little like Bill Belichick. I'm sorry. But to say it, but we have to be aware that we're right 90% of the time when everything I've said, it's not just a one-off explanation. It's something that's been dealt with. So Paul looks like he just popped his computer or camera on. Did you have a question? No. Okay.
More of appreciation of the joke.
Believe me, I didn't write that one ahead of time. Okay. Well, thank you for taking the time. And
We want to say thank you very much to Professor Danny Welch for this webinar presentation and taking the time to speak to our Pivot audience. It's very much appreciated.
thank you. Thank you all.
You.
Thank you. Bye-bye.
Bye.
Let's adopt